science fans, and welcome to SHE-ensya. Our topic for today begins a whole new series on genetics and molecular biology, starting with biomolecules. There are four types of biomolecules that are also known as macromolecules because of their size. However, for this video, we will be only focusing on 3 out of the 4. Each type of biomolecule can be found in different food items we consume, some in higher proportions than others. First, let's talk about carbohydrates, the nightmare of most people on a diet. What is this molecule all about? One of the primary functions of carbohydrates is to serve as fuel to make sure our cells function. No other molecule can be immediately processed by the cell to create ATP, the molecule that powers all of our cells and makes sure they function. The process of converting glucose to energy is facilitated by our cells' mitochondria. Since carbohydrates form a common part of our diets, some of us actually think that it is easy to break down and digest. But you might be surprised to think that some complex carbohydrates are some of the toughest molecules around. The major component in the rigid cell walls in plants is a carbohydrate known as cellulose. Cellulose is a linear polysaccharide polymer with many glucose monosaccharide units. The acetal linkage of cellulose is beta, which makes it different from starch. This peculiar difference in acetal linkages results in a major difference in digestibility in humans. Humans are unable to digest cellulose because we do not have the necessary enzymes to break down beta-acetal linkages. This makes cellulose still an important part of our diet because they serve as fibers that help clean our digestive tract. But what about the cows? Animals such as cows, horses, sheep, goats, and termites have symbiotic bacteria in the intestinal tract. These symbiotic bacteria possess the necessary enzymes to digest cellulose in their GI tract. Let me just clarify, however, that these organisms themselves do not have the enzyme to break down cellulose. It's the bacteria that's present in their stomach that is able to do so. No vertebrate is capable of digesting cellulose on their own. Another function of carbohydrates is linked to their ability to form linkages with proteins on the surfaces of cell membranes. This allows them to create unique identity molecules that allow our immune system to identify which cell is which type. One example of this is in our blood. The surface of our red blood cells have identity molecules in the form of carbohydrates linked to proteins, also known as glycoproteins. These are important because our antibodies screen the cells going around the body. And if they do not recognize the glycoproteins on the surface of the cells, they will attack them as if they are foreign cells. Carbohydrates, together with proteins and nucleotides, are what we call chemical polymers, which means they are made up of repeating subunits called monomers. The monomers of carbohydrates are called monosaccharides. Monosaccharides can be classified based on the number of carbons they have. There are 3 types of 3-carbon sugars called aldose, ketose, and triose. 4-carbon sugars are called tetroses, and 6-carbon sugars are hexoses. Also, carbohydrates don't usually have linear structures, but instead exist in a cyclic conformation. Monosaccharides alone can be functional already, such as glucose, fructose, and galactose, which are natural sweeteners found in our food. But in order to make other forms of carbohydrates, linkages must be formed between monosaccharides, and this is done through the creation of glycosidic linkages. Let us now move on to another biomolecule, to proteins, the favorite biomolecule of bodybuilders.
Muscle! Muscle! Muscle!
most important functions of protein is as messenger molecules that provide instructions to different body parts on how they should react and perform these functions. Proteins can do this as neurotransmitters or as hormones. Both neurotransmitters and hormones are messenger molecules in our body that allow our bodies to react to stimuli. The difference, however, is how the information or the chemicals carrying the information are sent to the receiving cell or organ and the speed of the effect. And finally, the more popular function of proteins is in making up the muscle present in animals. This occurs through our body breaking down consumed protein into amino acids, and these amino acids are eventually used to create more muscle fiber. And speaking of amino acids, there are 20 different kinds of amino acids. They are usually classified into 4 groups that affect the characteristics and functionalities of proteins that contain them. 5 out of these 20 amino acids are charged. At pH 7, 2 are negatively charged and are therefore acidic, while 3 are positively charged and are in turn basic. Aspartate and glutamate are the acidic amino acids, while lysine, arginine, and histidine are basic. The degree of the side chain's acidity or basicity is dependent on their charge. Here, you can see that aspartic acid is slightly more acidic than glutamic acid, while arginine is the most basic. The arginine side chain is very basic because its positive charge is stabilized by resonance, while the 2 nitrogens of the histidine side chain have a relatively weak affinity for H plus ions and are only partly positive at neutral pH. Regardless of pH, these charged molecules are considered polar and are therefore hydrophilic, which means they like water. These kinds of amino acids will not have any issues traveling around parts of the body that are rich in water or are aqueous. When proteins are created, these amino acids are often found on the surface of proteins and help form ionic bonds with neighboring molecules. There are 5 more polar molecules and are also hydrophilic. However, these amino acids are uncharged. Their polarity comes from uncharged amino groups or hydroxyl groups, and these amino acids are usually on the outside of proteins and are frequently engaged in hydrogen bonds. Out of these 5 amino acids, though, cysteine is considered special. Some amino acids have special properties, such as cysteine, that can form covalent disulfide bonds to other cysteine residues, glycine, that is more flexible than other amino acids because of its size and simple side chain, and proline, that forms a cycle to the polypeptide backbone. And finally, we get to our hydrophobic amino acids. These amino acids tend to cluster together and often stay inside protein structures. Unique from this group of 8 is tyrosine because its aromatic side chain makes it tend towards hydrophobicity, but the hydroxyl attachment balances that out a little, so it can get classified into different groups. Earlier, I mentioned that some amino acids can be found outside the protein while others are found inside. Why is this the case? Well, because proteins have different levels of structure where the primary involves the amino acids linking together, the secondary and tertiary is made up by the protein folding upon itself based on the amino acids it contains, forcing some of the amino acids to be inside and others on the outside. And finally, quaternary is when 2 or more proteins form complexes together. But regardless of what final structure the protein finally turns into, a protein only begins to form when 2 amino acids form a peptide bond from a condensation reaction. Yeah, proteins can be called peptides too. And finally, we move on to the third and last biomolecule for this video, lipids.
Is butter a carb? Yes.
The most important function of lipids is in each of the 30 trillion cells in your body, and it's in the structure called the lipid bilayer. Specialized fat molecules line the surface of the cells and allows for the perfect mix of fluidity and rigidity. Lipids can also function as hormones. Remember hormones? Molecules that are deposited into the bloodstream and cause changes in our bodies. If you remember correctly, proteins can be hormones too. Their difference is that proteins tend to be water-soluble, and in order for them to cross the cell membrane, which is made of lipids, they need receptors to ferry them across. Lipids, as molecules, are more resilient than proteins. This causes them to stay in the bloodstream for much longer and create long-term effects. Proteins, on the other hand, are more short-lived and create quicker effects. By the way, lipids are not true polymers because they are not made up of repeating subunits. There are two classes of lipids, your glycerol lipids and also your steroid lipids. Steroids are lipids because they are hydrophobic and insoluble in water, but they do not resemble traditional lipids since they have a structure composed of 4 fused rings. Cholesterol is an example of a steroid lipid. It is the precursor to vitamin D, estrogen, and testosterone. It is also an essential molecule in our cell membrane because it helps stabilize the structure. However, excessive consumption of cholesterol can cause fat buildup in our blood vessels that can lead to heart-related diseases. A glycerol-based lipid, on the other hand, has a glycerol backbone, shown here in green, with as many as 3 fatty acids attached to it, shown in red. The lipid in your cell membrane is a glycerol-based lipid that is special. This is because it's confused. A portion of it likes water or is hydrophilic, while another portion is classically hydrophobic. Why is this so? This is because instead of having 3 fatty acids attached to its glycerol backbone, it only has 2. The spot for the third is occupied by phosphate, which is polar and likes water. These kinds of molecules are called amphipathic. There are 2 kinds of fatty acids that can attach to the glycerol backbone. Saturated fatty acids only have single bonds and are stiff, while unsaturated fatty acids have double and even triple bonds, making them bendy. Unsaturated fatty acids, or the bendy ones, are healthier for us, and this is because they do not solidify easily and have lesser chances of solidifying inside our arteries and veins. But wait. Fans of the ketogenic diet, I think, would be arguing at this point that I missed an essential function of fats, which is that they are sources of energy too. Yes, that is actually true. In fact, carbohydrates, proteins, and lipids can be all considered energy molecules because they can be converted into prerequisites that allow cellular respiration to happen inside the mitochondria. The ketogenic diet is a very low-carb, high-fat diet that drastically reduces carbohydrate intake and replacing it with fat. This reduction in carbs puts your body into a metabolic state called ketosis. Ketosis is a metabolic state characterized by elevated levels of ketone bodies in the blood or urine, which is a byproduct of the body transforming lipids into metabolic fuel. The ketogenic diet, however, is a fairly new trend, and excessive consumption of fat and proteins can lead to pancreatitis, liver problems, kidney stones, stunted bone growth, and so much more. It can also trigger ketoacidosis, which occurs when the body starts breaking down fat at a rate that is much too fast. The liver processes the fat into a fuel called ketones, which causes the blood to become acidic. And apart from its other symptoms, it can actually be fatal. A healthy and balanced diet is still the best choice because there is no quick and easy way for you to get fit fast. If still in doubt, it is best for you to consult with professional dietitians rather than relying on fads found in the internet. I hope you were able to learn something from our short video on biomolecules. If you did, please give this video a thumbs up. Please subscribe to our channel for more science goodness as we pursue more information about genetics and molecular biology. If you have any comments, questions, or suggestions, please don't hesitate to message me, your resident Filipina scientist, in the comments section below. Thank you very much, and see you around!